I'd like to introduce Dr. David Stelly. He holds joint appointments with Texas A&M University and Texas A&M AgriLife Research. Dr. Stelly received his Bachelor's of Science in Plant Breeding and Cytogenetics, or his Bachelor of Science in Plant Breeding and Genetics from the University of Wisconsin-Madison, his Master's degree in Plant Breeding and Cytogenetics from Iowa State University, and his PhD in Genetics from the University of Wisconsin-Madison. Dr. Stelly focuses mainly on the improvement of upland cotton, as well as the development of a platform for the wide hybridization of sorghum and the creation of new energy crops. His research is largely geared towards optimizing the use of naturally occurring germplasm through such means as wild species germplasm introgression, reproductive cytology and genetics, and genomics. Thank you. So let's give Dr. Stelly a warm welcome.
Thank you very much, Sarah. It's a real pleasure to be here. I studied here from 76 to 79. Thanks to a nice tornado that came by my field when I was in the middle of my degree, I was able to stay here an extra year. It was actually quite an experience. It went about a half mile away from the agronomy farm where I had 80,000 F2s out there for my project, and they're about this, after it went through, they were about this high, and there was nothing else left except for that stem. That was it. So anyway, a lot of other students come through here have that privilege of extending their educational process an extra year, and I actually benefited a lot from it. So I'm really happy to be able to come here and talk today about some of the things that we've been working on and to share some philosophical perspectives and also relate some information as to both some of the successes, challenges, and opportunities that we're looking at today. So I'd like to start off by thanking the graduate students who've put in so much effort to organize this symposium. I was listening to Tabari. I was kind of reminiscing a little bit from conversations both before I came and then after I got here. There's a little bit of a cult getting going amongst the graduate students at different institutions, and Tabari didn't mention this, but a number of them are starting to watch the symposia at the other institutions, and I think that's kind of an interesting evolution in the process of the symposium system that's been developed. And I'd like to also congratulate and thank not just Tabari, but DuPont Pioneer for their support. Obviously, that's been instrumental to all of the symposia, including this one. I'd like to mention Kendall Lemke, who's just up here, and the department for their support, obviously, and specifically three of the students that so far, anyway, have been particularly helpful to me, in particular, Sarah Jones, who just introduced me, Matt Javits, and Maria Betza Mantilla. So thank you very much. I would next like to take just a moment, if you allow me, to just say a word or two about Reed Palmer and Holly here. So when I was studying here, there were, one of the best things of this place actually is the people. And I say that generally. I really do think that the people here are just a tremendous asset. But these two individuals for me were particularly valuable, and I wasn't able to attend either of their funerals, but I do very much value the things that they gave to me. So I just wanted to mention that. So, what I'd like to do is move forward and present just a bit about sort of the philosophical context of what we've been working on and give a little bit of a sense to everyone, but especially the students, about what it is that we're doing, why we're doing what we're doing, what's working, what isn't, and take what you can from that. So for myself, I guess the philosophical context of this is I see that we're in a pretty serious fight, if you will, to both provision society and also address the needs for some economic benefit to people, that comes through agriculture. And yet, at the same time, we have to deal with the fact that we do have limited natural resources. We have to preserve our environment. We need to have sustainability. I think this has been pretty grossly undervalued to date. Obviously, has gained a lot of momentum, and I think it will continue to do so because there's so many areas of the world where agricultural footprint has really been quite extensive, and I don't think we're even close to seeing how bad that can be in specific areas. On top of that, we have major biotic challenges and abiotic stresses that we really need to do a better job of addressing. It's not to say we haven't been doing it, we just need to do more. Some of these figures around the diagram kind of underscore some of the issues: extensive irrigation, depleting aquifers, et cetera, runoff, agricultural runoff leading to, or promoting at least, algal blooms, even in the seas around our shorelines, and in some cases extending extensively out into the seas. It's not just the U.S., it's around the world. New diseases coming in, such as greening in the U.S., for example, drought effects and all the water challenges that we face, major diseases that some of which are both long-lasting and in other cases very toxic. That's a picture of some Fusarium spores. There's a new rose disease, for example, that's come into the U.S., the rosette disease, that we're going to have to contend with. And to address all of these things and yet supply our food, feed, clothing, structural materials, and all the other many things that come from plants, really that's sort of the area where plant breeding comes into play. And in order to address these things from a genetic standpoint, we need to have genetic variation. And so that's where I have directed most of my attention, both in terms of introducing genetic variation. Obviously, we need to identify what parts of that genetic variation are variable, and then we need to be able to actually use it, which really, for me, we primarily work in the area of interspecific introgression. And when you bring in genes from an alien source, you can pretty much bet your booties that right next to it is a gene that you don't want. So that means we really need to have ways of separating the wheat from the chaff, separating the good from the bad, and then being able to use the good for the benefit of the breeding process. So another point I think that gets left off the chart in terms of government policy way too much is a recognition that plant breeding is really a translational science. If you think about it, what we do in plant breeding is take the scientific produce and put it into a product. And so institutions like NSF that purport to be really moving this process along need to be more engaged in the plant breeding, with the plant breeding community and really helping this process, because if we're going to do the research, we should be making use of it and getting all the bang for the buck that we put into it. So I think this is a very important aspect that has gotten undervalued in the political scene. Another point that I consider very important, it's kind of like banking. Plant breeding doesn't entirely, but we do recoup much of the previous progress. And so we're accruing genetic benefits every kind of breeding cycle. And so, yeah, it's not 100%, but we obviously see this progress. And so it's kind of like compounding interest on a bank account. Hope you have a bank account. And the benefit to society, obviously, is very significant. If you look back historically at the U.S., much of a good chunk of this economic growth in the U.S. over the past 150 years plus, about 150 years now, really kind of relates extensively to the amount of money that's been available for things beyond food, right? So with the improvements that came into the research and education and extension process from Lincoln onwards, we've seen a progressive increase in the ability to produce more and more product with less and less labor, less and less cost, and that money then basically translates into being available for other applications, and those stimulate your economy. So, coming back to cotton, which I'm going to speak on mostly today, the challenges and the opportunities and so on that exist for cotton are largely either identical or very similar to those which face other commodities and other crops. You know, the kind of the unusual things are that we have to deal with the fiber properties, and those are competing with a synthetic fiber market. So they're competing internationally with synthetic fibers for the textile processing. That's the major use of the crop, even though the seed is a pretty valuable oil seed. It's a major foodstuff for ruminant animals. And of course, there are a lot of other kinds of products. But like the other crops, we have some major kinds of issues, such as depicted in this little cartoon down at the bottom, that need to be addressed. And of course, that's where the breeding comes in. So again, my main kind of areas of work have to do with, one, bringing about the transfer of genetic diversity for the purposes of breeding, and then creating tools and knowledge and so on that enable more effective use of that germplasm. And I kind of look at this as involving three different aspects. One is the idea that when we get down to it, we're really, complex traits really don't have a very good specific understanding of what's behind those traits. We kind of mess around in this black box, and we bump into things that seem to be pretty important, and then we take it into a different environment or different genetic background, and a lot of times it doesn't work quite the way we expected. And so we're still trying to grapple with that. Secondly, when we do find valuable genes, we have to combine them together. Finding them could be difficult because they could be, in some cases, constrained by recombination processes and things of this sort, or interactions that don't work the way we want. And lastly, there are specific genetic hurdles that we have to deal with, as if we specifically deal with when we're trying to transfer genes, for example, from wild species to cultivated species. And I'll talk a little bit about that. So moving on with cotton, everybody in this room is going to be familiar with the idea of gene pools. So in the case of cotton, and I'm going to talk about this a little bit more, we can think of the crop cultivars as occupying that little circle inside the yellow one in the middle. Then we have the primary germplasm, which I'm going to describe in a moment, which goes beyond just the cultivated species itself. And then the secondary germplasm, which includes some diploids. And so I'm going to talk about those in a little bit more detail. The idea being the further away you get from the center of this diagram, actually the cultivated material, the more difficult it's going to be to use that germplasm. So in the case of cotton, we can identify these primary and secondary gene pools that I'm going to describe in a little bit more detail as being related to these genome groups that are defined in Gossypium. So Gossypium occurs in all of the tropical and subtropical areas of the world, but those are somewhat different from each other depending on where you look more closely. The cultivated Levi type cotton, for denims and so on, which is kind of considered a lower grade cotton, it's still quite good, is just one of several tetraploid species, what are regarded as tetraploid species, more about that in a moment. And then the rest of the species, there's 4 more identified and a couple of recently identified other species that are also at the tetraploid level. And then the balance, which is about 45 species, actually are diploid species. And if we look at their distribution, all of the tetraploid species occupy this primary gene pool, and then around it are some species that come from the New World, the D genomes, and then the A, B, and F genomes are found in Africa and Asia. So this is a listing, just totally, you don't need to memorize those. Let's just give you an idea of the relative numbers of species in these different gene pool groups. So the cultivated cotton's evolutionary origin looks something like this. It included some ancestors that are thought 1 to 2 million years ago to have formed an ancestral polyploid, a single polyploid, that gave rise to a number of different tetraploid species. And so that's up at the top. And one of those tetraploid species was ends up being the cultivated cotton today, along with the $100 shirt long staple cotton, or Pima cotton as we call it here. It's also the same as Egyptian cotton, which is this lower yielding but has a longer, finer fiber. Anyways, the origin of this ancestral polyploid involved two different genomes, which are most closely related to the A and the D genomes. Those are from Africa, Asia, and the New World, respectively. And sorry, I got my numbers wrong there. 7 to 8 million years ago was the formation of the tetraploid. Then 1 to 2 million years ago was 7 to 8 million years ago was the divergence of the A and the D that gave rise to the tetraploid 1 to 2 million years ago. So why is this important? Well, today we actually have 4 cultivated species, and 2 of those are the 2 tetraploids that I mentioned already, the $35 shirt cotton, the $35 pants cotton, Gossypium hirsutum, and the $100 shirt cotton, the Gossypium barbadense, and then 2 diploids that occupy very minor acreages in relatively high stress environments. So let's talk about the cultivated hirsutum cotton. Again, this primarily is for Gossypium, the Levi cotton, but also to some extent very closely related barbadense. So there are 52 chromosomes in this species. They're depicted here for Gossypium hirsutum. The genome is actually pretty modest in size. It's a little bit smaller than corn. So it's about 2.5 to 2.6 gigabases. So basically each chromosome is something on the order of 100 million base pairs on average. So you can see that today we have 13 pairs of A genome chromosomes, 13 pairs of D genome chromosomes, which basically means it looks like a deck of cards. It's a nice way to look at it. And the relative sizes of these 2 genomes is actually depicted here. So the A genome chromosome actually is about twice the size of the D genome. So the question is, however, based on some work that we did in the 1990s, whether or not a better analogy might be something more like a pinochle deck, where you actually have a higher level of ploidy than we normally think about. So we talk about cotton as being a tetraploid, and so yeah, that reflects a relatively recent polyploidization event. But based on a few lines of evidence in the 1990s, including a couple of fish-based approaches where we were working with some bacterial artificial chromosomes and with linkages to the nuclear organizers, as well as with specific repetitive sequences. Both of these actually gave us very strong visual evidence that we actually had at least one higher level of polyploidization that could be visualized. And then very strong evidence came out of the RFLP work, primarily led by Patterson's laboratory, which was published in '94 by Reinisch et al., that actually, if you look at the RFLP fragment number that you get, it's not 2, which is what you would expect from a disomic tetraploid, but 4, which would be more what you would expect from an octoploid. And sure enough, more recently, there's some really super elegant work that's been done using genome sequencing. This comes from a paper that was put out in Nature, again by Andy, and it shows multiple levels of polyploidization. And so in the diagram on the right, there's actually evidence of very, very ancient hexaploidization down at the bottom of the diagram. And then as you go up, you can see additional levels of polyploidization. And the one that's most recent then is the one that's shown up near the top, the 2-fold ploidy increase. And so that's the one we're talking about being a tetraploid. But you can see it's up over on top of everything else that's happened evolutionarily. So that explains actually why cotton is such a bear to work with from a SNP development standpoint and things of that sort, and we'll get to that in a moment. But from a breeding standpoint, one of the things that has driven us is that if we look at the cultivated species, and primarily I'm talking here about Gossypium hirsutum, so this is part of the primary gene pool. If we look at this diagram, it's kind of an old study, it's done with SSRs, but it's nonetheless very telling, and there have been lots of other studies that basically say the same thing, but this one suited my purposes here. On the right side, you can see this this band, very dark band on the right side. So those are all the different elite lines that have been genotyped and then run through this clustering development and printed out in this graphic dendrogram. And up at the very top, I've expanded this, and you can see for some comparisons. Sorry, my mouse doesn't show on the laptop. So this group up here actually shows the end of the cultivar type materials, and then at the very top we see a race stock, which would be a wild type, so it's a little bit further out. And then at the very top over here, you can see all the rest of them going out, way out here. And those are actually for a couple of other, quote, tetraploid species. So what this does is it kind of shows you the uniformity amongst the elite materials, which are on the very far right, some divergence from the race stocks, which would be representative, if you will, of the rest of the hirsutums. It's not quite right, but pretty close. And then very far away, the other tetraploid species. So what this is describing is basically the primary gene pool from left to right, and it's not really tapping at all into the rest of the Gossypium species. It doesn't include any of the diploid materials, which are much, much more diverse. So anyway, we've taken upon ourselves to, over a number of years now, to work with the tetraploid species and trying to introduce that germplasm, and more recently we've been focusing a little bit more on embracing some of the diploid materials as well. What I'd like to do in this slide is allude to one of the more important features of being a polyploid is that you tend to have a higher level of tolerance for various kinds of aneuploidy. And particularly important in this is a tolerance for hypoaneuploidy. And so, because hypoaneuploidy, it turns out, is a very powerful tool for doing many kinds of genetic analysis and genetic manipulation, some of which I'm going to talk about in a few minutes. One of the things that happens in these is, and this is depicted on the bottom part of this diagram, is that the tolerance for hypoaneuploidy is kind of graded. So you can have a species that's intolerant, won't tolerate any kind of hypoaneuploidy. Then you can have some tolerance in the sporophyte, the plant itself, right? But the question then is, will you have any kind of sexual transmission or not? And the general rule is that you will get maternal transmission in many instances, but no pollen transmission. And then you have other species that basically could be tolerant of both. And so I've just put a few examples here. So you can have maize plants which are actually hypoaneuploid. These are typically derived using the RX1 radiation mutant. Cotton is kind of a little bit more tolerant than that. The plants are pretty good with being hypoaneuploid, and they will transmit to some extent through the female gametophyte that hypoaneuploid condition. Sugarcane would be a little bit more so. It's, of course, typically a little bit higher ploidy and more extensively aneuploid as well. So this hypoaneuploidy actually provides us this opportunity to do a number of different kinds of things. So we developed some conventional cytogenetic stocks, and we've used those for a number of different applications. And these include the breeding, as shown on the right, the breeding of breeding interspecific F1 hypoaneuploids, and then using those as a stepping stone towards the breeding of chromosome substitution lines, as well as for various genomic mapping purposes. And I want to talk a little bit about the chromosome substitution because that has led to an important platform for us. So since many of the students here haven't done any cytogenetics, I actually threw this diagram in here, teaching opportunity. On the top is shown a euploid, an individual that would have a normal chromosome representation. So in this case, 26 pairs of chromosomes. Below that, I show different constitutions of hypoaneuploids. The first one being the monosomics, which actually have just, for one of the chromosomes, only has one representative rather than two. And then just below that are the monotelodisomics. People don't like the term telosome anymore because there's always a telomere at the end of the chromosome. So technically those are acrocentrics, but the idea is that you've basically deleted all or most of an arm. And below that are tertiary monosomics, which are also deficient for one chromosome, but basically carry a translocated chromosome, so it's missing pieces of two different chromosomes rather than one entire chromosome. Above and beyond that are the segmental aneuploids. Oops. Oh, not sure where that happened. Uh-oh. What am I doing wrong? I can just click this, I guess. I'll just use that. Sorry. So this is just to give you an idea of what they actually look like. The plants are actually reasonably robust. They do have morphological changes, somewhat like a Down syndrome in humans, although that's a trisomy, not a monosomy. But there's a syndrome of phenotypic consequences that result. Each one of these would probably involve a single copy then for about 500 to 4,000 genes. We've been able to develop aneuploids, hypoaneuploids for most of the 26 chromosomes, either with entire chromosomes or parts of them. And this turns out to be a fairly valuable resource. Using these, we can implement them into a backcrossing program. So we use them as a recurrent parent, as depicted here. And we create a hybrid. So that shown here is a backcross 0 F1 hybrid. So it'd be heterozygous for all of the chromosomes except one. That one would be basically only have the alien genes, and so that's actually a very nice way to identify, for example, all the SNPs on that particular chromosome. You can compare that to the other ones and localize all the SNPs that correspond to it. And then as you backcross repeatedly, identifying each generation your hypoaneuploid until basically you're diluting out your donor germplasm for all of the chromosomes except for the one that's being maintained as a hemizygote. And because it doesn't have a pairing partner, it's going to remain intact. And then when you get to the generation that you want to stop, you say that's enough, you inbreed, and then you identify the true breeding type. So this is an idealized version of a chromosome substitution depicted for Gossypium barbadense, for chromosome 25, that's been introduced into Gossypium hirsutum. So again, this would probably include about 500 to 4,000 alien alleles, and it would be quasi-isogenic with the recurrent parent and quasi-isogenic with every other chromosome substitution line that's developed in the same sort of manner. And these are true breeding lines. They're easy to plant and so on, so they're very suitable for, in most cases, very suitable for doing replicated experiments. And so what we've been doing is developing these and then establishing collaborations in order to evaluate various traits, because there's no way we can evaluate all the traits that are of interest, especially if you need to look at different environments. By doing this, you're essentially reducing the genetic noise when you're doing comparisons, because most of the first order and second order interaction effects have been removed. So they're a very, very nice tool. We've actually created a number of these, and these are the chromosome numbers are depicted here. for 3 different species, all of them tetraploid, Gossypium barbadense, the $100 shirt cotton, and then a Hawaiian wild cotton called Gossypium tomentosum, and then one from Brazil called Gossypium mustalinum. And some of these are still in progress. More recently, all of these were developed, by the way, with conventional methods, and so now we're actually in the process because, as I'll mention in a moment, we've developed some very nice SNP resources and SNP techniques. We're going back to analyze these in more detail. We've done some preliminary work with SSRs, and we know that a few of these are kind of messed up, and there could be problems that arose from handling from a human standpoint, and or there can also be some biological causes for that. But anyway, we're starting to address that. But anyway, this platform, however, is very important, and here I've depicted the kinds of families, for example, that you can create and use, and the kinds of analysis that you can look at. And again, these were done using more or less conventional methods, and we're in the process now of bringing in the SNP analyses as part of this process, and of course it'll get a lot more detailed as a consequence. But just for example, if you take the chromosome substitution lines themselves, you can look at the genetic components that contribute to the phenotypic variation. If you go into the F2s, you can look at break it out into the additive versus dominant type of components. You can take it to multiple generations, start looking at epistasis. Quite interestingly, when you look at top crosses, you start to see specific interactions between the alien chromosome and specific cultivar genotypes. It's more effect there than I would have anticipated, but it's interesting. Then you can make intercrosses between the chromosome substitutions. Again, you can look for epistatic interactions, gene-gene interactions that come into play. And of course, one of the more fertile areas for that would be to take homologs that have been brought in from one species and look for complementary factors that are segregating on those 2 chromosomes. And we'll kind of expect to find that to be more prevalent than for other chromosomes. And then lastly, I'm going to illustrate this in a moment. Much as has been done in wheat, you can create chromosome-specific rills, which are kind of nice because you can get some very nice resolution on your QTLs. This is just an example and a chance, I guess, to talk a little bit about cotton quality scoring. For those of you who don't work on cotton, it might be an interesting thing to look at. So, of course, we've got lint yield, and you've got seed cotton yield is both the lint and the seed together. Then we run the seed through a gin to separate the lint. So we look at lint yield, and of course the lint is the most valuable, and it typically runs about 30% of the seed cotton weight. And so then people look at the percent of lint. And so the typical value here would be 30%, but it can run much higher. And so this is an area where breeders will operate a lot, and it has a quite high heritability. Then boll weight is pretty obvious. Micron error is basically measured in terms of how fast air will move through a given sample. And so it's kind of a measure of thickness in the fiber. It's more than that, but it's related to that. Then this is a measure of the shorter wave or short fiber content, the elongation, and then the strength. So the strength is obviously pretty important when you're doing textiles. And I circled this one just to illustrate the fact that if we look at this particular trait with the F2s, you have about 60% of that going into dominant source of variance. So it's kind of interesting. Other traits were more balanced out. I'm quite interested in the lower part of this because it allows us to get into the epistasis, and so we're just kind of moving into this area. We've had a couple of studies that we've published in this area, but I think generally this is where the most interesting results are going to be coming out. And so as you start to work with more advanced types of crosses, you're going to get more into the detail. So I said I was going to talk about the rills for just a moment because I think this is an interesting kind of illustration. Shown on the top here would be a conventional type of, would be the parents, and rill number 1, rill number 2 follow the idea that rill number 1 would be homozygous, 4 different bits and pieces. roughly 50% of each parent, but randomly, well, I shouldn't say randomly, but homozygous for each of the particular segments. And then rule number 2 would also, but of course it would be different segments from rule number 1, but you're segregating for genes on all the chromosomes, whereas if we're talking about a chromosome-specific rill developed with a idealized line, we're basically just creating rills that are being segregating for a specific chromosome. So you can imagine that the phenotypic, genotypic correlations and so on would be much higher in terms of the information it provides about this chromosome. So with 30 to 50 lines, you can do a really pretty powerful analysis of this chromosome. And so that's one of the areas that we're moving into pretty rapidly. We've done a little bit of work on this so far, and more coming down the pipeline. In fact, we've released some lines based on those analyses. So what are we doing with the line research? We're moving towards fuller characterization of all the lines. We're really starting to push on the chromosome-specific rills because of the power they give us in evaluation, particularly in concert with SNP data. And so we're looking to define high-resolution QTLs through this method. And then also to work more and more with the chromosomes chromosome substitution intercrosses to get after epistasis and so on. So one of our big challenge right now is to integrate the SNP methods into this process. Let me jump through this slide. There's a slide produced by a former student and postdoc, and it just gives sort of a timeline of where we've been in terms of SNP development. We really started working seriously on this around 2007, 2008. And so the first sort of SNPs were just coming out about that time, and through some pretty big efforts, in 2014 we released a high-density SNP chip on Illumina platform. It's all Infinium 2 assays, and those have been run across a number of different things. So in total, there are about 63,000 good assays on that. So you have about 90% success with the assay development, which is pretty typical. And of those, 45,000 are intraspecific. That is, they're amongst different elite lines of hirsutum. And we put an emphasis on that in order to drive the marketability of the chip, because the biggest users of this chip will be commercial users. And so we needed them to buy the chip in order to bring the prices down to where other people that work in public arena could actually use it. And this is done in consultation, by the way. So we developed a nice international consortium to come up with a plan. I fought pretty hard because I work in the area of interspecific introgression. I fought pretty hard to include a decent representation of interspecific SNPs. And so you can see here that we have close to 18,000 interspecific SNPs. And by that, what I'm talking about is they're polymorphic between the wild species and hirsutum. So that for us was much more important. They're really not designed to find polymorphisms amongst different wild species per se. And then there's still room for add-on content on this chip, so a private company can stick their own SNPs on it if they want to. So once we developed the chip, we ran it across large numbers of samples, and we established basically 38,000 genotypes on this that work quite well and can be automatically genotyped using Genome Studio software. So we ran across these samples in order to do that. We didn't use all of them to develop. The cluster file is really developed for working with the tetraploid material. So I'd like to, before I move on, so one of the ideas now is we're kind of at a convergence of this chromosome substitution platform development and SNP development, so we're just now trying to put those things together. I'd like to, sort of for the last part of the talk, turn my attention to another project which is quite different in nature and I think instructive. It's not totally successful in some ways, but it's been an interesting project. And this involves an effort, collaborative effort. A lot of our work is collaborative, but for Reniform nematodes, which have gotten to be a pretty big problem. You can see down in the bottom here a depiction of the presence of this disease. Actually, this is from 2001. It's quite a bit significantly worse now. It's a fairly important problem and can be quite devastating, particularly in the southeast. So, interesting, the best source of resistance that was described for this pest is Gossypium longicalyx, which is an F genome species. So we're going outside of the primary gene pool. We're going into the secondary gene pool. This is an African species, totally wild, doesn't produce any cotton. Here's a depiction here in the center here of what it looks like. So a hybrid was made between that and Hirsutum. A triploid was created that was chromosome doubled using colchicine, and then it was crossed with a Mexican species, which is a D genome, to create a synthetic trispecies tetraploid with a genomic constitution of FADD. That's the way I talk about it, is FADD. And in this hybrid, you'll see in a moment, it doesn't, actually the pictures I show you doesn't show it that well, but basically the F genome can pair with the A genome to some extent, and the D genomes will pair quite extensively with each other. So anyway, we use this trispecies hybrid in a modified backcross program. I'll show you a picture in a moment. And then after we went through that backcross program, had inbred development, inbred extraction, and we actually released a couple of lines out of this. So this is what the pedigree looked like for this. It's pretty much of a god-awful mess. And this was largely, this was put together by Forrest Robinson, who used to work over at the USDA in close concert with Dr. Al Bell, who's a plant pathologist with the plant pathology group over there. And so we had a really nice team together. I had a graduate student that helped with a number of things in this, and we basically got involved from the genetic analysis and cytogenetic analysis standpoint, initially cytogenetically, and then with the genetics, I was really pushing on the markers. And so as a part of this process, we were doing the cytogenetics and trying to facilitate this process because we wanted to get back to the euploid level where we had normal pairing and things of this sort. And we found out that as we went through the backcross process, we were getting back to the 52 chromosome level pretty quickly. So you see this gets to 100% by the BC3, so that was important. However, when we look at these, we found that there were two motile types. One of them would form 25 bivalents plus 2 univalents. And so one of these is the alien F genome chromosome, and one is the A genome, corresponding A genome chromosome. And then there were others where we actually had 26 bivalents. But even so, that doesn't necessarily mean this is totally correct. Anyway, so the idea behind the interpreting these two differences was that if you have no crossing over between the longicalyx chromosome and the hirsutum chromosome, you're going to end up with 2 univalents. And if you do have one or more crossovers, then you can end up with a bivalent. If you have it on both sides, like here, then you're going to have what's called a ring bivalent. Oops. Another thing that came out of this is when we tabulated the data, it looked like we were getting somewhere around 10% recombination between those two chromosomes. So it was an impeded level of recombination, but workable. Okay, we could do something with that, assuming it was random, pretty much randomly distributed relative to the normal recombination. Another thing that we did in relationship to that pedigree and some ad hoc families was develop some linkage relationships. And in this case, we've got a couple of markers that showed pretty good linkage with the resistant and the susceptibles. You can see the differences in the banding patterns. So we were able to localize the marker, and very quickly, initially with some resistance, but pretty quickly converted the pathologists and nematologists over to using the marker-assisted selection route, and then from there, basically, this became really the first marker to be, or at least one of the first markers to be extensively used in cotton for marker-assisted selection. And this just shows some linkage relationships. So I mentioned that we released some lines, and we actually shared them with a bunch of people before we even released them, in order that they might grow out these lines and start breeding with them and testing them and so on in different locations. And it wasn't too long before we got back some nasty reports saying that your resistant lines are not doing too well. And so as you can see in the diagram here, this is actually the resistant line versus the susceptible line. And what happened was, if you grow the lines in a field that does not have high nematode populations, it does well, and if it has no nematodes, it does really, really well. But if you grow it in a fields that have high nematode, at least some fields that have high nematode populations, they're very highly stunted. And so there are a number of different populations as to why that could occur. We hadn't seen it because we had grown our cotton after corn or sorghum, and both of those will kill the nematode. And so we hadn't grown it in high population soils, and so it was kind of interesting revelation for us. So we decided to tackle this problem to see if, by chance, it's hopeful thinking perhaps, but that there might be some sort of, the little roller is very sensitive on the mouse, that there could be another locus nearby that affects susceptibility to soil-borne pathogens that in high nematode soils are having a field day with this material. If you remember, for example, Fusarium race 1 gains entry into roots through root knot nematode, and so there was some question of, well, maybe there's something similar going on. So we decided to go in and do some more analysis in this, and we went in to work with SNPs that we developed from some RNA-seq data, a few of which were on the chip. Anyway, so we identified large numbers of SNPs, and then we use information about these and the available sequence data to pick out SNPs that would be in the area of interest, and did some analysis of these using CASP assays. And to extend this, what we wanted to do is develop a protocol where we could actually do marker-assisted selection to select for recombination events that were very, very close to the gene of interest, so this resistance gene. So we wanted to create and select for resistance or recombination events right next to the gene. And so to do that, we wanted to have a way of screening through large numbers of seed. We didn't have to grow the plants, and so we developed a non-destructive, really cheap seed DNA extraction method. Cost about $0.05 each. Seed do pretty well. You just throw some fungicide on them, and they do just fine after that. And we use this to screen large numbers of seed. So one of my students spent 3 weeks doing these extractions. This is all by hand, by the way. I'm sure it'd be a lot better if we were to automate it, but I think Monsanto could probably get on our case if we did that. Anyway, we did this manually. She extracted DNA from 20,000 seed and then ran markers that had been through a selection process and found 5 recombinants in this area. And we analyzed those recombinants, but unfortunately none of them gave us the separation between the stunting effect and the Reniform nematode resistance. But nonetheless, this screening technique and the markers actually, I think, it foreshadows sort of a new era in cotton breeding where we can do large-scale marker-assisted selection on seed or seedlings, actually, and really sift through large populations to find recombination events that are of value to breeding. So I'd like to wrap up at this point, probably even a little bit running late. As I get to the acknowledgments, I wanted to make just a couple of points. So just to summarize, one is, the polyploidy and the redundancy in the genome has massive effects on both the biology of the plant as well as the genomics. And so this has affected a lot of our operations. But it also created opportunities for doing cytogenetic manipulations that you can't do in, quote, a diploid species. So this chromosome substitution platform, which we're actually expanding through marker-assisted selection to include isogenic chromosome segment substitutions, is very, very powerful for quantitative genetics and trait gene association establishment. And at the same time, accomplishing breeding goals. And so it's really a multi-use type of platform. So this availability of a cheap marker-assisted selection technology, I think, is really going to be a transforming thing for us. We're putting it at the front end of a lot of our breeding cycles now. Before, we've been kind of occasionally doing marker analysis at the end of a cycle to see what do we have. We're putting it at the front end, and so it's going to make a big impact that way. And then kind of related to that is the idea that we've very successfully developed these SNPs and have a capacity to do high throughput, high quality genotyping using the SNP chip, but then at the same time, we can use that information and the maps that we've created out of that to pick out specific markers and then convert them over. We use the CASP assay a lot into individual assays that we can run individually or in moderate numbers on single plants or whatever we might choose. So that's kind of where we're at right now. And I'd like to acknowledge the people that have contributed. There's a couple different generations of our group, but it's a pretty international group. Kind of oscillates between having more men and women. Right now, recently it's been a lot of women, and they've done a great job. I've had a lot of really good collaborators as well. I'd particularly like to cite the Mississippi State USDA group for the chromosome substitution line. They've been great partners in that. And more recently, we've been roping in additional people as we start looking at additional traits. SNP development, I especially want to note Alan Van Dynes at UC Davis, and at that time, Dr. Hamid Ashravi worked with him, and I work a lot with Jeff Chen at UT on the genome sequencing. Sorry, the contrast here is not too good. Down at University of Texas on cotton genome sequencing. For the Reniform, I'd like to especially acknowledge the USDA group in College Station, including Al Bell and formerly Forrest Robinson as well, and Jose Quintana works with him. And then for the SNP chip, there's just too many people to mention. But again, Alan Van Dynes and especially the Australian group with CSIRO. With that, I think I'm going to stop. I was asked to talk about this mutant, but I don't have time. This is a semigammy mutant, which is actually a haploid chimera with male and female derived sectors. With that, I'll stop for questions. Or actually, I probably don't have time.
We'll take just a few questions while we get set up here.
Okay, great.
So just a reminder, if you do have a question, please use the microphones so that people on the webinar can hear. And those of you on the webinar, once you hear this, I know there's a bit of a delay, but please use the online question form that was on the webinar link, please.
So there's been a couple of recent papers that have talked about how our genes are using the cell cycle to perturb their defense, to take advantage of the cell cycle to initiate defense responses. Mm-hmm. Have you considered that maybe your resistance gene is actually doing that, and it's not unlinked?
Yeah, well, we have been considering that it might not be linked, but I mean, we had several hypotheses, and so the easiest one to attack was linkage, and so far it hasn't worked. So, yeah, I mean, the other hypotheses are still definitely out there. So, yeah, we're definitely thinking about that, and actually been looking at the hormonal system in general, jasmonic acid and all that. So, yeah.
Thank you for your nice talk. So my question is related to your CS platform. So in the primary population, for example, in F2 population, if the variance of epistasis is very small, it's very low, what the variance of epistasis could be in CS plant form in advanced populations. In other words, if the epistasis variance in F2 population is very slow, is it possible we can detect the significant epistasis QTL in CS plant form?
So for the epistasis, we've generally been going, we initially went to using F3s, F4s, and so on, but now we're really looking more closely at top crosses as being a way to look at, mainly because we can find useful things out of that at the same time, and so we've kind of been really seeking out analytical methods that also get us towards a breeding objective. And so top crosses are really nice that way. Johnny Jenkins actually kind of took the lead, well, he did take the lead on doing that initially, and we're actually doing some more now with Wayne Smith at College Station. For me, I'm really interested in both the chromosome-specific rills, and actually Sukumar Shaha at Mississippi has been taking kind of the lead on that. I mean, we're pushing forward together on it, but he's taking the lead. And then we're kind of both working on the chromosome substitution by chromosome substitution crosses, which is interesting. If you think about it, if you do F2s, you got everything integrated at once, and so you have all the interaction possibilities are feasible. If we do a chromosome substitution by chromosome substitution, you give up most of that opportunity, but you get a lot more precise information because you've eliminated all that noise. It's kind of like being a Geiger counter in a lead-protected room. You know, it'd be much easier to find a source in that case as opposed to being in a high radiation environment. So I think from an analytical standpoint, you almost want to attack a problem from both directions, one using the very complex method and one using a very simplified model system approach.
Thank you.
Yeah.